Hello friends, I am MD Harun Al Rashid and it's my YouTube channel Harun Biojon. Today in this video I will discuss about the structure and role of phytochrome, a plant photoreceptor. The first, what is phytochrome? Phytochrome and what is their role? Lights play a vital role in plants for develop different physiological process like photosynthesis, plant growth and development, and others factor like seed germination, seedling deutylation, leaf expansion, stem elongation, phototropism, stomata, chloroplast movement, shade avoidance, circadian rhythm, and flowering. All these process are regulated by light. But here the question is, which molecule of the plant perceive light? This is the question. All physiological processes are controlled by light, and which molecules or which pigment absorb this light? This is the main question. And all this process is maintained by circadian rhythm. Circadian rhythm means changes of different physiological process by environment in plants. Okay, this is called circadian rhythms. Okay, circadian rhythms is maybe daily process or seasonal process. Photomorphogenesis is how plants respond to light signal by growing and developing. Photomorphogenesis is a part of plant biology that determine or that represent the role of light signal that develop the plant morphology. Yes. Red and blue light are very powerful in triggering photomorphogenic response in plants. The different growth and development process of plants is regulated by light, and this light, and especially red light and blue light, mainly regulate all this process. The proteins pigment that control photoperiod detection was initially referred to phytochrome. Okay, phytochrome. So, which molecules that perceive light? That is phytochrome. Phytochrome means phyto means plant, and chrome means color. Plant color. So phytochrome is a bluish pigment that receive light and help in different physiological process like germination, deutylation, inhibition of stem and petal elongation, chloroplast development, chlorophyll biosynthesis, leaf expansion, branching, flowering and senescence process. এই সমস্ত সব প্রসেসগুলো রেগুলেট হচ্ছে কার দ্বারা? phytochrome পিগমেন্টের দ্বারা. There is another pigment that also controls this process is called cryptochrome. Okay, that detect mostly blue light. Photoreceptor and circadian clocks are universal mechanism for sensing and responding to the light environment. Okay, so what is circadian clock? Circadian clock means the environmental clock that regulate the physiological or morphological process in living organism. Circadian clock regulate daily activities or seasonal regulation. Okay, daily activities like photosynthesis, seasonal regulation like flowering process. The phytochrome is a bluish biliproteins pigment responsible for perception of light and absorb red light and far red light. Phytochrome absorb two different lights, that is red light that absorb 680 nanometer of light or 660 nanometer of light, and this is the PFR that receive 730 nanometer light. PR is an inactive form and PFR is the active form. Okay. So who discovered phytochrome? Phytochrome pigment was identified by spectrophotometers in 1959 by biophysicist Werner Butler and Harold Sizelman. Okay. And Butler also named this pigment and exists in two intercompatible forms. The first discovered by Warren Butler and Harold Sizelman in 1959, and they Warren Butler also named of this pigment, and he says that there are two forms of phytochrome, that is PR form and PFR form. PR is an inactive form, biologically inactive form, but PFR is a biologically active form. PR receive 660 nanometer of red light, but PFR or far red light receive 730 nanometer of light. This PFR is required for flowering in short day plant, but PFR is required for the flowering of long day plant. Okay. PFR is always associated with hydrophobic areas of the membrane system. PFR always associated at the hydrophobic region of the membrane system, but PR is found diffusible state of cytoplasm. PFR always found in the hydrophobic region of cell membrane, but PR is found in the cytosol. The interconversion of two forms of phytochrome is mainly involved flower induction. So why interconversion is important? Because these two forms help to flowering in two different type of plants. Like PR helps in flowering in short day plant, but PFR inhibit flowering in short day plant. But PFR stimulate the flowering in long day plant. So what is the difference between PR and PFR form? PR is a red light is a blue green in color, but PFR is light green in color. PR is inactive forms of phytochrome and it does not show phytochrome mediated response, but PFR is active form of phytochromes and show biological responses. PR is a maximum absorption 680 nanometer, but in case of PFR, maximum absorption is 730 nanometer. It can be converted into PFR form when absorbs 660 nanometer of light. PR absorbs 660 nanometer light and converted into PFR, but PFR absorbs 730 nanometer of light and converted into PR form. PR is diffused throughout the cytosol. PR present in cytosol, but PFR found in the hydrophobic region of cell membrane. The PR form contains many double bonds in pyrrole rings, but PFR forms only double bonds in all pyrrole rings. So what is the role of phytochrome? Especially phytochrome role play in flowering process. During daytime, PFR accumulate in the plant. During daytime, PFR accumulate in the plant, and this PFR is during daytime high concentration of PFR present in plant. So this high concentration of PFR stimulate flowering in long day plant. During critical dark period, short day plant this form, but during dark period, this PFR converted into PR form. So during dark period, the PR concentration in plant is high. This PR condition is helpful for flowering in short day plant. Reversal of the inhibitory effect of the red light during critical. So this is a short day plant. This is dark period and this is light period. Okay, when dark period is less than light period, no flowering occur. But when dark period is more in than light period, the flowering occur in short-day plant. But if we apply some red light during this dark period, no flowering occur in short-day plant. Okay, if we use some red light. Okay, red light, no flowering occur. In case of long day plant, this is the short night and long day flowering occur, but short day, long night, no flowering occur. If the dark is breakdown by use of PFR light, then flowering occur. Okay, so PFR is the helpful for flowering in short long day plant, and PR is helpful for flowering in short day plant. Clear? And PR builds up floration activity in short day plant, but PFR activate floration in long day plant. So what is the structure of phytochrome? Phytochrome has been detected in wide range in plants in angiosperm, gymnosperms, bryophytes and algae. Okay. Dark ground seedling are richest source of phytochrome. Normally phytochrome found in all type of plants, but etiolated dark seedling, that is, this is dark seedling, is the richest source of this phytochrome pigment. In 19 and also especially found in the apical meristem of the plant. In 1983, Virastra and Quayle successful purification of intact native phytochrome from etiolated oat seedling, which is soluble protein. Phytochrome is a soluble protein and first isolated by Virastra and Quayle in 1983. It is 250 kilodalton protein. It is a homodimer. It is 125 kilodalton and this is also 125 kilodalton protein. This phytochrome has detected in different parts of seedlings within the cell. Phytochrome exists in nucleus throughout the cytosol. Each polypeptide has prosthetic group called chromophore. This is chromophore. This chromophore is synthesized in plastid. Okay, this is chromophore and synthesis in plastid. And this chromophore is bind with the disulfide bridge of cysteine residue with the polypeptide chain. This is the protein part of phytochrome. This is non-protein part of prosthetic group of the phytochrome, which is this protein part and non-protein part combined to produce the hollow protein. This chromophore part or prosthetic group part is found in the plastid, but this protein part is found in the nucleus. And these are joined by the ether bond of cysteine residue. Okay, assembly of these two component phytochrome is autocatalytic, and autocatalytic means this is the enzymatic kinase enzyme activity are present in this phytochrome. Phytochrome has tetrapyrrole ring 1, 2, 3 and 4. 4 pyrrole rings are present. Difference between these two forms is the 15 number carbon, that is this. This is form, but here this is a trans form. This is PR form, this is PFR form. This is the tetrapyrrole ring containing phytochromes, and this tetrapyrrole ring is similarly structured with phycobilin, so it is called phytochromobilin. There are two major types of phytochrome in plants, that is type 1 phytochrome and type 2 phytochrome. Type 1 phytochrome is predominant and found in green plants and seeds, but type 1 phytochrome is encoded by phytochrome A gene, while type 2 is encoded by phytochrome B, C, D and D, Y and E gene, phytochrome E gene. Clear? And this phytochrome type 2 is found in green plants and seeds, but phytochrome A is mainly found in etiolated seedling plant. The other mechanism of phytochromes is phytochrome. The mechanism of action of phytochrome is not clearly known, but it may be act by the ATPase activity, regulating by the ATPase activity, by controlling the activity membrane-bounded hormone gibberellin. It controls the activity of gibberellin and modulating the activity of membrane-bound proteins. Or by regulating transcription of numerous genes involving multiple signal transduction pathway. All these are the mechanism of action of phytochromes. So overall, the role of phytochrome is it controls the seed germination process, detiolation process, inhibition of stem and petiole elongation, chloroplast development and chlorophyll biosynthesis, leaf expansion, branching of plants, flowering of plants and senescence. All these processes are controlled by this phytochrome. I think you understand this whole process and it is beneficial for you. But thank you for watching this video. Thank you again. Now we discuss about another pigment, bluish pigment, that is cryptochromes. These cryptochromes is found in all plants and animals both. Okay, the cryptochromes in plants are blue light receptors. They receive or perceive blue light like blue ultraviolet A sensors. Contain FAD, flavin adenine dinucleotide, that regulate vital function. Clear? Photomorphogenesis like seedling growth and leaf expansion, circadian rhythm, that means internal clock or morphological and physiological changes according to the environmental condition, day and night condition. Okay, flowering, response to stresses acting via photoreduction and phosphorylation by interacting with signal partners, control genes. All this process like photomorphogenesis, circadian rhythm, flowering, response to stress are controlled by cryptochrome genes. Cryptochrome pigment. Cryptochrome is a blue light receptor. Okay, it also helps in germination to fruit, seed germination. In Arabidopsis, there are two types of cryptochromes are found: cryptochrome 1 and cryptochrome 2. Cryptochrome 1 and also another type of cryptochromes are found, that is cryptochrome 3. Cryptochrome 1 and 2 primary photoreceptor for development, photomorphogenesis and flowering. The cryptochrome 1 and cryptochrome 2 is a primary photoreceptor that helps in photomorphogenesis. Photo means light, or morphology of the plant. Also, in the presence of light changes the morphology, like leaf expansion, like seed germination, like flowering, like circadian rhythm control, etc. process. Okay, and cryptochrome 3 in function in mitochondrial and chloroplast DNA repair. So what is the key function of cryptochromes? For photomorphogenesis, means inhibit the hypocotyl elongation, promote proper seedling development. That means this is a seed and after germination the seedling is developed. Okay, this is called hypocotyl. Circadian rhythms. Circadian rhythm means internal clocks maintain. Enter the plant internal clock, the day-night cycle crucial for daily process. Flowering, regulate the photoperiodism. On the basis of day length, the flowering occur in plants, and on the basis of this day length, the plants divided in two types, that is short day plant, long day plant, and also another type is day neutral plant. Stress response involve adapting drought, salt and high light condition. Also the another key function of cryptochromes. Other sources like stomatal opening, root development, apical dominance and pathogen responses are also controlled by cryptochrome genes. Here we discuss the structure and mechanism of action of cryptochromes. The cryptochrome is a blue light receptor. Okay, this is the structure of cryptochrome. So in cryptochrome there are two domains, that is PHR domain and CCT domain. PHR means photoliase homology region. Okay, this is the conserved across species in the all organisms. This is conserved or similar type, essential for light perception. The PHR photoliase homology region is essential for photoreception or light perception, contain alpha and beta domain and form a pocket-like structure. This is the pocket for FAD, flavin adenine dinucleotide, that act as a chromophore component. Chromophore component in cryptochromes. Okay, binds FAD and absorb blue light, initiating signaling. The PHR or photoliase homology region is conjunct for different species. Okay, it has two domains, that is alpha domain and beta domain, and form a pocket-like structure that binds the FAD. FAD is a chromophore of cryptochromes. Another domain is CCT domain, that is C-terminal domain. Okay, this is N-terminal. PHR is N-terminal, but CCT is a C-terminal domain. Variable in length and sequence. In different organism, the length is different in different organism. CCT region, functioning the signaling effector domain, undergo light-induced conformational changes affecting interaction with downstream protein like COP1 and SPA1, which regulate gene expression and development. COP1 means this COP1 is a constitutive photomorphogenesis 1 and SPA1 is a suppressor of phytochrome A1 gene. When this SPA1 and COP1 interact with CCT domain, the cryptochrome is inactivated. Okay, in the presence of blue light, this SPA1 and COP1 is inactivated and active the phytochrome genes. Clear? Phytochrome genes to regulate growth, circadian clock, and flowering also. So when this SPA1 and COP1 gene is inactivated in the presence of blue light activates the HY5 gene, that is called the hypoglottal elongation gene. Elongated hypoglottal 5 in plant encode a crucial BGIP transcription factor acting as a master regulator for light responses, photomorphosis, and coordinate growth, development, and nutrient used by controlling thousands of downstream genes. When this SPA1 and COP1 is inactivated, the cryptochrome become phosphorylate in the presence of unknown kinase X. Then activated the HY5, HFR1, and MYB-LFF1 gene. These are transcriptional factors that transcripts the mRNA, produce different type of protein, and show the responses. The structure related to DNA photoliase. This is a photoliase, PHR, that is photoliase region. Undergo conformational changes of blue light absorption. যখনই blue light absorb করলো তখন কি হচ্ছে COP1 and SPA1 inactivated and activate the phytocryptochrome genes, leading to a signal transducing through its effector CC domain. C-terminal extension of cryptochrome protein, which are photoreceptor in plant. How it works? First, light absorption. Okay, light absorb like blue light and excite the FAD molecules within the PHR domain. यहाँ पर এখানে যে FAD presence রয়েছে, when light absorb, the FAD remain in oxidation condition. It convert into redox condition and change its conformation. Then conformational changes occur and trigger structure and PHR domain affecting the interconnecting loop influence CCA domain. Then CCA domain activated. What is Signal transduction. The activator cry interact with the target protein like COP1. When blue light absorb, FAD convert into FADH and the target protein like COP1 and inhibit this COP1 and SPA1 gene expression and shows the gene expression and development process. Oligomerization. Photoactivate and cryptochromes can form homotetramer, a structural state. Homotetramer, a structural state crucial for the signaling function and CC domain. Okay, CC domain structure. CC means what is CC domain? CC domain means chromosomal domain. Chromosomal domain, that is activate the DNA and response the genes. Major function is circadian clock regulation, plant development like photomorphogenesis by activating the HY5 hypocotyl elongation gene 5, flowering process by activating the florigen, stomatal control that crucial for gaseous exchange, and magnetoreceptors means proposed to sense the earth magnetic field. Some species through this it is still under investigation. Gravity, they perceive the gravity or magnetic force of earth. I think you understand the cryptogram. So thank you for watching this video. Thank you again. If you like it, please like, share and subscribe my channel to your friend. Thank you.